Hey there people, Jay here, and in this video I'll briefly explain to you about the process and enzymes involved in DNA replication. Mainly there are three different kinds of enzymes that are involved in the process of DNA replication. First one is nucleases, second one is polymerases, and third is ligases. So let's talk about each of them separately. Nuclease enzymes. The nuclease enzyme acts to hydrolyze or break down a polynucleotide chain into its component nucleotides. A polynucleotide chain that is held together by 3' 5' phosphodiester bond is broken down by this enzyme. The nucleases are of two types: exonucleases and endonucleases. Exonuclease enzyme begins its attack from a free end of a polynucleotide chain. Therefore, depending on the specificity of the enzyme, an exonuclease will either begin at a free 3' OH site or 3' phosphate end of a polynucleotide chain. In both the cases, the enzyme travels along the chain in a stepwise manner, liberating single nucleotide monophosphate and eventually digesting entire polymer. Endonuclease enzyme also attacks one of the two sides of phosphodiester linkages, but they react with those bonds that occur within the interior of a polynucleotide chain. If the polynucleotide chain is a single stranded, as for example viral DNA, such an attack will obviously cut the chain into two pieces. But however, if the polynucleotide strand is a member of a DNA helix, which are mostly found in prokaryotes and eukaryotes, a single endonucleotide cut will create a nick in the helix. This nick can also serve as a substrate for an exonuclease. This created nick likely interrupts the hydrogen bonding between bases in the vicinity of the nick and eventually affecting the limited untraveling of the helix. Second enzyme, and very important enzyme, that is DNA polymerase, synthesizes one polynucleotide chain that is copy of another. Both prokaryotes and eukaryotes contain this DNA polymerase. Three different DNA polymerases are known in prokaryotes, of which DNA polymerase 1 and 2 are meant for DNA repair, while DNA polymerase 3 is meant for actual DNA replication. So DNA polymerase 1. This enzyme was isolated by Arthur Kornberg, this guy, in 1960s, and this was the first enzyme that was suggested to be involved in DNA replication. It is also called Kornberg enzyme. This DNA polymerase 1 is now considered to be a DNA repair enzyme rather than a replication enzyme. This enzyme is known to have 5 active sites, namely template site, primer site, 5' to 3' and 3' to 5' cleavage site or exonuclease site, and nucleoside triphosphate site. This enzyme is mainly involved in removing RNA primers from Okazaki or precursor fragments and filling the resultant gap. This enzyme can also remove thymine dimers, which are produced due to UV radiation. Scientists have done much X-ray crystallography to look at the structure of DNA polymerase. In fact, most of the work has been done on a fragment of a DNA polymerase called Klenau fragment. Klenau fragment or polymerase was initially prepared by cutting the natural E. coli DNA polymerase 1 into two segments with a protease. One of the segments retained the polymerase and 3' 5' exonuclease activity but lacked the 5' 3' exonuclease activity. This Glenau fragment is shaped like a cupped right hand, with enzymatic activity taking place into two places, separated by a distance of about 2 or 3 nucleotides. It is proposed that when the polymerization site senses a mismatch, the DNA is moved so that the 3' end enters the exonuclease site, where the incorrect nucleotide residue is then cleaved. Polymerization then continues. DNA polymerase II resembles DNA polymerase I in its activity, but is a DNA repair enzyme. It brings about the growth in 5' to 3' direction using free 3' OH groups. DNA polymerase III plays an essential role in DNA replication. It is a multimeric enzyme or holoenzyme having 10 subunits such as alpha, beta, epsilon, theta, tau, gamma, delta, delta dash, chi, and psi. All these 10 subunits are needed for DNA replication. However, all these subunits have different functions. For example, three of the subunits, that is alpha, epsilon, and theta, form the polymerization core with both 5' to 3' polymerase activity and 3' to 5' exonuclease activity. Another subunit, that is beta subunit, is a processiotic clamp. This beta subunit forms a dimer or a donut shape around the DNA so it can move freely on the polynucleotide chain. Okay. So let's brief about the properties of different polymerases. All three polymerases contains 5' to 3' polymerization as well 3' to 5' exonuclease activity, while only DNA polymerase 1 has 3' to 5' exonucleases activity, while polymerase 2 and 3 do not. Polymerase 1 requires single-stranded primers and can use nicked strands, while polymerase 2 and 3 cannot. The affinity of NTP precursors of polymerase 1 and 2 is lower compared to polymerase 3. Eukaryotes, for example, yeast, rats, human tumor cells, are found to contain the following types of DNA polymerases. DNA polymerase alpha is a high molecular weight enzyme and is called cytoplasmic polymerase or large polymerase. It is found in nucleus and cytoplasm. DNA polymerase beta is also called nuclear polymerase or small polymerase and is found only in vertebrates. DNA polymerase gamma is called mitochondrial polymerase and is encoded in the nucleus. DNA polymerase delta is found in mammalian cells and is pCNA or proliferating cell nuclear antigen dependent enzyme for its DNA synthesis activity. DNA polymerase epsilon was previously known as DNA polymerase II. This enzyme is pCNA independent and occurs in mammalian HeLa cell line and budding yeast. The large DNA polymerase alpha is the predominant DNA polymerase enzyme in eukaryotic DNA replication and was believed for a long time to be only enzyme that was involved in the process. But now one more polymerase, namely DNA polymerase delta, is also found to be involved in eukaryotic DNA replication. DNA ligases. These enzymes are capable of catalyzing phosphodiester bond formation between 3' free OH and free 3' phosphate groups, often nicked DNA, which was created by any endonuclease, thereby restoring an intact DNA duplex. Many DNA ligases ligases have been discovered. The ligase enzyme from E. coli requires the presence of NAD plus as a cofactor, while ligase enzyme from T4 bacteriophage requires ATP to bring about the joining reaction. Other proteins or enzymes involved in DNA replication. These following enzymes or these three types of proteins are needed to help the DNA double helix to open and provide exposed DNA template for the DNA polymerase to copy. First one is DNA helicase. These are ATP dependent unwinding enzyme which promotes the separation of two parental strands and establishes replication fork that will progressively move away from the origin. DNA helicases hydrolyzes ATP when they are bound to a single strand of DNA. Hydrolysis of a DNA, hydrolysis of ATP can change the shape of a protein in a cyclic manner that allows the protein to perform its mechanical work. DNA helicases utilizes this principle to move rapidly along a DNA strand. When they encounter region of a double helix, they continue to move along their strand, thereby unwinding the helix. Unwinding of the template DNA at replication fork could in principle be catalyzed by two helicases acting in concert, one running along the leading strand and another running along the lagging strand. Single strand DNA binding proteins or SSBPs. Behind the replication fork, the single DNA strands are prevented from rebinding about one another by the action of single stranded binding proteins. These proteins bind to exposed DNA strands without covering the bases, which therefore remain available for the templating process. Topoisomerases or gyrases. The action of a helicase introduces a positive supercoil into the duplex ahead of the replication fork. Enzymes called topoisomerases relaxes the supercoil by attaching to the transiently supercoiled duplex, nicking one of the strands and rotating it through the unbroken strand. The nick is then released. Thus, a DNA topoisomerase can be viewed as a reversible nuclease that adds itself covalently to a DNA phosphate, thereby breaking a phosphodiester bond in the DNA strand. Because the covalent linkage that joins a topoisomerase to DNA retains the energy of the broken phosphodiester bond, the breakage reaction is reversible. This resealing is rapid and does not require additional energy input. However, the rejoining mechanism is different from the mechanism of DNA ligase. One type of topoisomerase 1, causes a single strand break or nick which allows the two sections of DNA helix on either side of nick to rotate freely relative to each other, using the phosphodiester bond in the strand opposite the nick as a swirl point. Second type of polymerase 2, forms a covalent bond to both strands of DNA at the same time, making transient double strand break in the helix. DNA polymerase, RNA primases, and helicases may be associated with one another to form a multi-enzyme complex called the rectisome that carries out the leading, synthesis of leading and lagging strand in a coordinated manner. Likewise, the proteins at a replication fork cooperate to form replication machine. That is, the primase molecule is linked directly to the helicase to form a unit of the lagging strand called a primosome, which moves with the fork, synthesizing RNA primer as it does. Let's talk about the process of DNA replication in prokaryotes. The initiation of DNA prokaryotes involves recognition of origin followed by opening of DNA duplexes, DNA captures, which acts as primase activator. Alright, as I told you, the process of initiation comprises of three steps: recognition of origin or ORIC, opening of DNA duplex to generate a region of single-stranded DNA, and capture of DNA or 5' to 3' helicase, which acts as the activator of primase enzyme. Thus, DNA or the initiator protein and ATP complex binds at 9 base pair inverted repeat regions of ORIC in E. coli and promotes the opening of the DNA duplex in the region of 3 direct repeats of 13 base pair sequence called 13-mers. The opening occurs from right 13-mer leftward and requires negatively supercoiled DNA and HO or IHF initiated proteins. DNA bit or helicase is transferred to exposed DNA strand and causes unwinding of the DNA in the presence of ATP, SSB proteins and DNA gyrase. This results in unwinding of DNA duplex and the replication from ORI proceeds in both directions, that is bidirectional. Single-stranded binding proteins binds on single-stranded regions and two DNAB complexes, that is primosomes, are loaded one on each strand. Elongation. Elongation of elongation of DNA replication requires helicases, primases, DNA polymerase III, SSB proteins, RNase H, and ligases. As helicase or DNA-B travels in 5' to 3' direction, it generates a replication fork by opening the DNA duplex. The DNA strand having helicase becomes the lagging strand. DNA primase associates with DNA-B helicase, forming the primosome, which synthesizes multiple primers for lagging strand and single RNA primer for the leading strand. For the synthesis of lagging strand, DNA polymerase III has to work on the same strand which DNA-B or helicase is bound, but it travels in opposite direction. DNA-B helicases, DNA-G primases, and DNA polymerase III work together in strand elongation. Helicase and DNA polymerase assembly remains processive, that is, they remain tightly bound to the fork and stay bound through the reaction. Synthesis of lagging and leading strands take place by somewhat different methods. It is far more complex for lagging strand than for the leading strand. First, let's talk about the leading strand or continuous synthesis on leading strand. In bidirectional DNA replication, the leading strand is primed once on each of the parental strand. The RNA primer of the leading strand is synthesized by RNA polymerase. DNA polymerase III causes elongation of the leading strand, and finally DNA polymerase I and ligase enzyme give final touch to the leading strand, as in case of the lagging strand, which we will talk about now. Primase is taken up from solution and activated by helicase or DNA-B to synthesize a RNA primer, that is 10 to 20 nucleotide on the lagging strand. The RNA primers are recognized by DNA polymerase III on the lagging strand and are utilized for synthesis of precursor or Okazaki fragments. In fact, each new RNA primer is recognized by gamma subunit of DNA polymerase III and loaded with alpha subunit of the same polymerase. This preloaded beta subunit may then capture the core of DNA polymerase III holoenzyme when it becomes available after finishing its synthesis job on the preceding Okazaki fragments. On completion of the Okazaki fragments, the RNA primers are excised by DNA polymerase I, which then fills the resulting gap with DNA. After DNA polymerase I adds the final deoxyribonucleotides in the gap left by the excised primer, the DNA ligase forms the phosphodiester bond that links the free 3' OH end of the primer replacement to the 5' phosphate end of the Okazaki fragment. Okay, this was the process of DNA replication in prokaryotes. In DNA replication in eukaryotes, similar mechanism is followed, but the enzyme that are involved differs. Eukaryotic DNA replication requires two different DNA polymerase enzymes, namely DNA polymerase alpha and DNA polymerase delta. DNA polymerase delta synthesizes the DNA on the leading strand, whereas DNA polymerase alpha synthesizes the DNA on the lagging strand. Besides this enzyme, six more factors are involved. T antigen, replication factor A or RFA, also called RPA or eukaryotic single-stranded binding proteins, topoisomerase 1, topoisomerase 2, proliferating cell nuclear antigen or PCNA, also called cyclin, and replication factor C or RFC. Before the onset of DNA synthesis, there is a pre-synthetic stage of 8 to 10 minutes duration for the formation of unwounding DNA complex. This step needs only three proteins, namely T antigen, RFA, and topoisomerase 1 and 2. The T antigen, using its DNA binding domain, forms a multi-subunit complex with the site 1 and 2 in the presence of ATP and causes local unwinding. More extensive duplex unwinding occurs due to association of RFA and topoisomerase 1 with the help of DNA helicase. Topoisomerases help in unwinding DNA by altering topology of DNA at the replication fork. RFA or single-stranded binding proteins bind to unwounded DNA strands. The primer RNA synthesis is performed by primase, which is tightly associated with DNA polymerase alpha. DNA polymerase alpha helps in synthesis of Okazaki fragments in 5' to 3' direction in eukaryotes. Okazaki fragments are much smaller, only about maybe 130-135 base pair longs. That is about the size of DNA on a nucleosome. Replication factor C and PCNA helps in switching of DNA polymerase so that polymerase alpha is replaced by polymerase delta, which is then continuously synthesizes DNA on the leading strand. Another Okazaki fragment is then synthesized from the replication fork on the lagging strand by polymerase alpha primase complex, and this step is repeated again and again till the entire DNA molecule is covered. The RNA primers are then removed and the gaps are filled as in prokaryotic DNA replication. Recently, the role of DNA polymerase epsilon in DNA replication has been stressed upon, so that three DNA polymerase alpha delta epsilon are now known to be involved in eukaryotic DNA replication. Scientists have proposed that DNA polymerase alpha might function at both the leading and lagging strands, whereas polymerase epsilon and polymerase delta are involved in elongation of the leading and lagging strands respectively. All right. So this was the process of DNA replication. Uh, do tell me your suggestions in the comments down below, and if you have any questions or doubts regarding the same, you can also comment it down below. And thank you for watching. More videos will follow which will be more explanatory and could be quite big compared to this shorter basic exploratory videos. So stay tuned, subscribe to the channel and hit the like button, and I'll see you guys in the next video.